Hey, Bio 2 students, Mr. Yachts. Hope things are going well. Just going to kind of continue on with some notes. So, as always, fill in your note packet and fill out the note review for me when we're done. Okay, so here's the deal. So, we talked about Mendel's laws and we talked about how he came up with the law of segregation, law of independent assortment that we'll talk about some more here down the road. But let's say we have, and we did some Punnett squares on this the other day, or actually yesterday, that let's say that we don't know what dad is or mom is. He could be big B, big P, or big P, little p. Mom, we know. This is like we did with the Punnett squares on Thursday, where we didn't know if what the one parent is. Can't tell because they produce the same thing. Well, you can do a test cross if you're doing like bacteria or fruit flies or flowers or whatever. So if I cross this with a known recessive phenotype and I get a 50-50, just like we did the other day, then I know he's heterozygous or she. And if I get all the same, then I know that is the other type. Now, that's fine and dandy, and it works well for plants and bacteria and fruit flies. Obviously, you can't do this with humans or whatever. So that's just basically what a test cross is when you run across that term. It's this determining an unknown dominant phenotype by crossing it with a known recessive. All right. With a lot of organisms, we can do that with, but not all. All right. So that's basically what that is, nothing more, nothing less, okay? This next concept, we're going to do a little fun activity on this end of this week. We're actually going to do flipping some pennies. It'll drive you crazy. It sounds like Yahtzee going on there. But we can take the laws of probability, so a little math. And Mendel's law of segregation and independent assortment reflects these rules of probability. So when you're talking about probability, when you're tossing a coin, the outcome of one toss has no impact on the outcome of the next toss, right? So each time I toss that coin, I have a 50% chance of heads and 50% chance of tails, okay? And that's very similar to what we talked about in meiosis. One gene segregates into a gamete independent of other gene alleles that are on different chromosomes. Yeah, we talked about how, like in Scandinavia, they all have blue eyes and blonde hair, but that's on the same chromosome. It's basically just talking about if I'm looking at one gene and I'm looking at a different gene on a different chromosome, it's 50-50 because they line up in the middle during meiosis and they split. Which one do I get? It's a 50% chance, okay? Now, we're going to take this one step further, okay? Let's apply something called the multiplication and addition rule, okay? Now, we're just looking at a monohybrid cross, one trait. So the multiplication rule states that the probability of two or more independent events, you may have learned this in math class, will occur together is the product of their individual probabilities, okay? So in other words, let's say, what is the probability in an F1 cross? What's the probability of getting a black-haired child and blue eyes? Okay, so you multiply, right? It's the segregation of a heterozygous plant is like flipping a coin. So each gamete has a half a chance of getting a dominant allele and a half a chance of getting a recessive allele, right? Obviously not true with homozygous. It's a 100% chance. But we're talking about heterozygous. I got a 50% chance of getting a big letter and a 50% chance of getting the lowercase letter, right? So you can see that. So let's do a Punnett square with that. So we're flipping coins. You'll do this in a lab. I have a half a chance of heads, half a chance of tails, half a chance. So I have but look at the deal. I'm looking at the different combinations. This is where it gets tricky. So this will help you in the lab. So if I look at this, I have a fourth of a chance of getting heads, a fourth of a chance of getting tails, and I have a fourth of a chance of big R, little r, or a fourth of a chance of getting big R, little r the other way. Now, I get that you're going to say, well, wait a minute, Mr. Yance. That's really a 1 to 2 to 1 ratio because these are the same outcome, big R, little r. However, we're saying in this case, head on this one, tail on this coin, tail on this same coin, heads on the other coin. If we're looking at the combination. So that is a little bit confusing, if that makes sense. It's actually a fourth of a chance for each of those combinations, even though two of them are the same. All right. Now, the rule of addition states that the probability that any one or two or more exclusive events will occur is calculated by adding together their individual probabilities. And again, this will make more sense when we do our little lab activity, but let's look at this. So the rule of addition can be used to figure out the probability that an F2 from a monohybrid cross will be heterozygous rather than homozygous. All right. So this is where it gets kind of a little bit crazy. Okay. So we can apply that and we can look at the whole probability. So let me just go over this and then we'll finish it up later. So let's put both of these together. Okay. So let's say I have multiple characters. And we'll do this. So let's say I have a dihybrid cross. I have multiple characters like blood typing or brown eyes, blue eyes, green eyes, hazel eyes. What's the odds of these occurring simultaneously? You get some crazy math here. So let's say I'm calculating the chances of different genotypes. You got to look at them as independent events and multiply them together. So let me show you. And I'll show you, and we'll go over it with a lab activity, and then when we get to the test, you'll be like, oh, yeah, okay, this makes sense. Okay, so look at this. This gets kind of crazy. So if we're looking at these different genotypes, we're saying, this is a trihybrid. So I got, what is the chance of an individual getting all lowercase and one heterozygous for a third trait? So these two traits are homozygous, heterozygous. It's a fourth times a half times a fourth, or times a half, and you get a 16th of a chance, okay? And you're looking at this saying, wait a minute, where do I get the half? Because you're crossing these two parents. We're looking at this as the outcome, but what is the probability of these two parents producing this outcome? Because this is a fourth of a chance because it's a half times a half, right? This one is only a 50% chance because you're only going to get a big Y or a little y because of the parent combination. This is a half a chance because of that. See how that works? It's kind of tricky. So you're only got a 16th. So if you add them all together, these two parents only have a 1/16th of a chance of getting this one, 1/16th of getting this one, but 1/8 of getting this combination. A little higher, right? Or 16. So you can see how this gets crazy. If you add it all up, what's the chance of at least two recessive traits? 3/8, right? If you look at that math. So it's a little bit tricky. I get it, and kind of confusing at times. But after we do the lab, I think it'll make more sense. All right. Okay, we're going to stop there. So fill in your notes, do the little note review. Really important, you put down questions if something doesn't make sense, and please ask them either on my paper if this is an assignment, or if I'm talking to virtual students, email me a question if something doesn't make sense. And hopefully that is clear as mud, right? But don't worry, we'll do some practice with it, and you'll be like, oh, this makes sense. It's kind of like it'll click in. All right? Hey, have a good day, and we will see you later. See ya. Bye.